Douglas Holtzman, Ph.D., MPH, Master Public Health, is the Chief Scientific Officer of Icosavax. He has 25 years of experience in industry and the philanthropic sector, leading and funding innovative research and development activities with an emphasis on vaccines. Prior to his position with Icosavax, Dr. Holtzman was Deputy Director for the Global Health at the Bill and Melinda Gates Foundation from 2004 to 2012, and Vice President of Discovery for Takeda Pharmaceuticals from 2012 to 2016. So, Dr. Holtzman, thank you very much for joining us. We've been looking forward to this for a long time, and I'll just turn everything over to you.
Okay, so I've unmuted myself. We got on early, so hopefully everyone on the call can hear me. So first, I'd like to thank Stan and Paul for the invitation. I mean, we're obviously at a very critical time in the evolution of the SARS-CoV-2 pandemic, and I'm happy to speak with you all today about a subject that I know is on everyone's minds right now, which is vaccines for the pandemic. And before we get into the formal presentation, I wanted to make just a few comments. First, I want to make it clear that I'm speaking with you today as an individual scientist that cares deeply about this topic and not on behalf of any organization, although I've already gotten some questions about the work that we're doing at Icosavax, and I'll be happy to share that. I've devoted the last 20 years of my life to vaccines for infectious diseases, and it's been really an incredibly rewarding to be part of efforts that have led to major improvements in global health. I was very fortunate to be part of the Bill and Melinda Gates Foundation's global health program in its early days, and I spent several years supporting efforts to improve global influenza pandemic preparedness. It was without a doubt the most interesting professional experience of my life, as pandemics land us squarely at the intersection of science and technology, economics, politics, and ethics, exactly where we find ourselves right now. So as Paul said, I'm currently the CSO, Chief Scientific Officer for Icosavax. It's a startup biotech company here in Seattle that's developing a number of vaccines, including one for SARS-CoV-2. And so you should be aware of that as I go through the talk. I'm going to try to separate facts from my opinions, but I am fundamentally pro-vaccine, and that will color my commentary. And then finally, I used a backbone for a vaccine presentation that I found online, and I edited it to serve for today's purposes. Unfortunately, I couldn't find who created the original, so I'm unable to thank them specifically for their contribution. And so with all that said, let's begin. I've left plenty of time. I only have about 22, 23 slides, so plenty of time in 90 minutes to cover the full presentation and to have lots of Q&A at the end. However, I'll also stop as I progress through the talk to make sure that the presentation is clear and to answer any questions you may have on specific topics that I'm planning to cover, which are the following. Has the slide presentation advanced?
Yes.
Okay, so we're going to cover the background and history of vaccines and pandemics in brief. We're going to talk about the stages of vaccine product development and also that the various technologies that are at play right now, because there's been a huge change in the vaccine landscape. And then we'll get into SARS-2 vaccines more specifically, including the status and issues with the current vaccines. And really, I think hopefully that's where we'll get into most of the Q&A that people have on their minds. So we'll start again with the background on a little bit of background on vaccines and pandemics. So what is a vaccine? A vaccine is any preparation intended to produce immunity to a disease, and vaccines include suspensions of killed or attenuated microorganisms or products and derivatives of microorganisms. So this is the definition, a definition from WHO. It's a little bit limited because it focuses on antibodies and humoral immunity, but the vast majority of vaccines protect through the generation of specific antibodies. We can talk, if we really get deep in the technology, we can talk about some of the other modes of protection that are out there, because they are issues that are on people's minds right now in modern vaccinology, including SARS-2. So I want to tell you a little bit about the history of vaccines, because I think it's incredibly interesting and important. So
let me interject for just a second. People, if you on these slides that have lots of verbiage on them, if you go to the top of your screens and look at something called view options, zoom ratio, you can zoom in a little bit on them and make it a little bit easier to read. Thanks, Doug. Sorry.
Okay, no problem at all. So I am in awe of the early pioneers of vaccinology who operated with only a really rudimentary understanding of pathogens and the intricacies of the immune system. I mean, these folks were able to create safe and effective vaccines before we even understood the central paradigm of molecular biology, which is DNA makes RNA makes protein. And Edward Jenner is really credited with the discovery of the first vaccine for smallpox, although vaccination may have started earlier in China. Is really thought to have been variolation was the first sort of approach to dealing with smallpox. And what he noticed was that milkmaids had beautiful complexions in a time when people had these horrible disfigurations from smallpox. And as he tried to understand what was going on, why were they protected when these epidemics were raging through, he noticed that as they were milking, that oftentimes they would have some scars on their hands that were the result of this sort of cowpox infection. This is turns out to be a related virus, and it provides protection against smallpox. And so what he did was rather extraordinary and incredibly dangerous, and would never pass an institutional review board in these days. But what he did was he infected people with this cowpox, and then after that, he challenged them with smallpox. He gave them the potential to have a smallpox infection and showed them to be immune. Now, again, as I said, we're actually going to come back to this at the end of the talk in terms of this concept of human challenge, because it's something that's being discussed right now for SARS-2. But nevertheless, the early history of vaccines is, the attempts to generate immunity by using attenuated organisms or killed bits of organisms, although recombinant DNA technologies have really changed the game for the development of new vaccines. Now, for those of you who are interested in this mid-century history, there are a number of excellent books about vaccines, and one of my favorites is called Polio, An American Story. And it's by an author by David Oshinsky, and it's a very accurate description of like the egos and academic competition that drove the development of the first two polio vaccines. It also covers the beginning of medical philanthropy with the March of Dimes, the Cutter incident, which was a big problem. It was a notorious incident where people were given a product that actually led to polio due to incomplete inactivation of the vaccine. And so this is one of my favorite books on vaccine history. And so I'd strongly recommend that for people who are interested in these sorts of issues and the history in particular. Okay, so if we go forward, the five most important attributes of a vaccine are safety, 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 and efficacy. Now, vaccines are given to healthy individuals. This is not a situation where someone's taking a drug for a cancer and you're willing to tolerate a whole lot of side effects. And so, scientists and clinicians are very, very sensitive to this issue. And of course, we'll come back to this because I think it's what's on everybody's mind, that given the accelerated development path, are these vaccines going to be safe and are they going to be effective? So I'll go to the next slide. And just to tell you a little bit about, how do vaccinations work? Again, I can't really do justice to the immune system. As I said in my email, any one of these topics could be like a full lecture, but in short, you have a remarkable system in your body that can recognize self from non-self and know to attack only these foreign invaders, including pathogens. Includes viruses, bacteria, worms, protozoa. And what vaccines do is they contain enough of the disease agent or protein to cause an immune response, basically to give you an exposure without getting sick. And the purpose of vaccination is to jumpstart the immune response and basically give you immune memory, what they call immune memory, because your body, once it sees a pathogen the first time, it's able to remember it. And then the next time it sees it, it's able to fight it off much more efficiently. Now, there's a lot of challenges with respect to vaccines. I mean, these are not easy to make. Certain organisms are particularly complicated, like malaria and HIV, TB. These things have been proven to be very intractable. I guess what I would say here just at the beginning, again, for a note of optimism, I am absolutely certain that we will have a safe and effective vaccine for SARS-2. I am a little less certain, and would be irresponsible of me to give you a specific date by which that vaccine would be available. But this is a pathogen that I'm very confident that the scientific community can handle using the tools that we have, and we'll talk about those, in sort of the third section of the talk. But vaccines have to be safe, as I said. They have to be able to be manufactured consistently. You have to be able to make it today, a year from today, and 10 years from today in exactly the same way. Because in the vaccine parlance, the process is often the product. It's not a case like with a small molecule or a drug where you can look at that small molecule with specific tools and know that it is exactly the same, that what's in the pill is exactly the same. Because these are complex biological products, it's the process that's important and the ability to monitor that process and reproducibly generate that process. Now, the other thing that's obviously a challenge for pandemics is you got to be able to, it's given to healthy people to protect against the possibility of future infections. And for the pandemic, we need billions of doses of vaccine. And so we'll touch on the manufacturing issues in the last part of the presentation. And so, but altogether, there's certainly challenges with vaccines. Now, I wanted to put this up front because I think this is, important, and it's a legitimate issue that people have on their mind, which is vaccine fears. And so, one additional challenge that vaccines face, though, is that they become a victim of their own success. Once they're effectively deployed and we have, broad immunity, the problem that they solve fades from memory, and the risks associated with vaccination become more problematic. And so, there are genuine risks associated with certain vaccines, although I will tell you that over the years that some of the problems have been addressed through the use of these new technologies that we'll touch on. But I've given you a couple of examples of problems that are known with certain vaccines. So, for example, measles, there's, 1 in a million or less than 1 in a million people can get a complication from measles vaccination. But, the death rate from measles, there's hundreds of thousands of people who still die in developing countries or in resource poor settings from measles if you're malnourished. If you're a young infant, people die from measles. We don't really realize that in the United States. And I have to say that, a lot of my focus was on the sort of international health rather than just, vaccines for the United States, through my time at the Gates Foundation. And so, this is, certainly it's a trade-off, but it's, in the big picture, we consider this an acceptable risk because of the damage that the measles virus itself can cause. And we've seen outbreaks in, Disneyland and in California when people stop using vaccines in, different parts of the country, even just across the Puget Sound here in Vashon Island, when people stop using vaccines, the diseases come back, and measles is an example of that. Similarly with yellow fever, these are live attenuated viruses versus some of the safer types of vaccines that I'll talk about. But even with yellow fever vaccine, which used to be a horrible disease and problem, about 1 in 1 to 2 million people will get disease symptoms from the vaccine. Now, a major myth, though, is this issue, which you've probably heard about, that vaccination causes autism. And this in this situation, I just have to be very blunt and say this is misinformation. There have been multiple studies that have disproven this. The person who promulgated this initially had his medical license removed, and the issues around the link between the measles, mumps, and rubella vaccination and autism are just simply a myth. I hope that I hope that that you all understand that in order for vaccines to be useful, they need to be used. And so this is one of the terrible things that this plague of this pandemic of misinformation that we're all dealing with on the vaccine front. It's a big problem, and it's actually one of the reasons why I agreed to present today, because I feel very, very strongly that as scientists we have to try to communicate clearly to the general public of what's really going on, what the real risks are with vaccinations, as I shared with you here, versus what the myths are. Okay, so now let's talk about pandemics briefly. Pandemics have been with us for a long time, and one point I really want to make to this group is that, and I want to make emphatically to everyone listening, is that you may have heard the comment that no one could have known we'd face a pandemic, and that is absolute nonsense. Okay, this is absolute nonsense. While it's true we couldn't predict exactly which pathogen would emerge and exactly when, as I said, I spent 5 years of my life working on influenza pandemics, and there are scientists who have devoted their entire careers to this issue. Now, unfortunately, sometimes they've been dismissed as chicken littles because of the unpredictable nature of pandemics. And we have to decide as a society how much we care about this issue, because it's not something that the private sector can manage on its own, because you can't build a factory and wait for a pandemic to hit to manufacture vaccines. It's really an insurance policy. And the one thing I can promise you is that this won't be the last pandemic we face. I mean, it's that I'm absolute certain. There are literally millions of viruses that are out there, and this has been done through deep sequencing and some of the molecular technologies that I've spent my life on. And as we've become the dominant mammal on the planet, it's really just a matter of time before evolution works its magic and selects viral variants that we've never previously faced that are capable of infecting humans. And that is essentially the definition of a pandemic. When these viruses have emerged and we've never seen them and we've never experienced them, and the entire human population is vulnerable. And I've just listed a few here. Everyone knows about the Black Death and in the Middle Ages, but before then there was the Justinian plague, also from Yersinia pestis. This is a bacterial infection that killed roughly half the population or a third of the population of the planet during the Justinian plague. Many people will be familiar with the Spanish flu that took place during 1918, 1919. There's been a lot of discussion of that in the news because it's got a lot of lessons learned. There are lessons that we can learn about, even the second pandemic wave that we're experiencing now as fall, came back from the earlier part of this year. And vaccination, I would just say, is a critical tool to fight pandemics. And we'll talk about, where we are and what's changed now that we've got newer technologies to tackle these. So I think that may be, so the last one again here, the last slide in this section, I think, is again a note of hope. I mean, we have been remarkable as a species in our ability to use science and technology to improve, public health. And one of the very best examples of this is the eradication of smallpox. This is a major, major achievement. And another book that I would point you towards, I was fortunate to work at the Gates Foundation with a man named Bill Melinda. He is a giant in public health. He was the first director of the CDC, and he wrote a book called House on Fire, which he's an incredibly modest man. He's just done, he's really one of my heroes, and I don't have too many of them. I mean, he's a genuine hero of global health. He's one of the humblest men I've ever met, despite his major accomplishments. And I would strongly recommend House on Fire as a sort of first-person history of what took place during the eradication campaign for smallpox. But we have the power use science and technology to minimize the impacts of these pandemics, and even to eradicate infectious disease with the right set of attributes for the pathogen, as well as the right focus. This was a WHO-led effort, and the whole world collaborated. Hopefully we can get back to that sort of global collaboration soon. Okay, so let me stop there briefly and see if people had any questions on that background section on history of vaccines and pandemics, and I'm happy to clarify anything if people want to. I guess, Paul, you're managing the Q&A. I don't know if people need to type it to you or because you've muted them.
I think we have a small enough group that let's try, if you want to unmute yourself and simply speak the question, that you can do that at this point. There aren't any typed questions in the chat right now.
Okay.
I have a question. This is Julie Palmer.
Thank you, Julie.
Screen's off. And maybe you covered this. I came on a little late. I apologize. Thank you for doing this presentation today. My question is, why are none of the Western countries' vaccine candidates killed virus vaccines, just traditional platform killed virus vaccines? Why are they all recombinant vaccines? I mean, I see China's doing just a traditional platform vaccine. I'm wondering why nobody in our sphere is doing it.
It's a very good question. I hadn't really. I mean, I think you're accurate. There may be. I think there may be a bias towards new technologies, and we will cover in the last section this issue of vaccine-enhanced disease. Now, at present, there's no real, there's not necessarily any reason to believe. I've not seen any data to suggest that vaccine-enhanced disease for SARS-2 is going to be a problem. But if it were going to be a problem, it would likely be a problem with with whole virus, chemically killed preparations like the one you're talking about in China, and that's also being developed for distribution in Brazil, I think. So because we have more cutting edge technologies, and that'll be the third section, I think people are biased towards recombinant approaches and synthetic vaccines, which we know to be safer because you don't have to worry about the, like I said, in the Cutter incident for polio, where they tried to inactivate the polio virus, but it was incompletely inactivated and people took the vaccine and got polio. So I think that as I describe the technologies, I may answer that question. But if I don't, I don't think I have a very specific answer. But I think it may be concerns about safety, sort of an abundance of caution on safety, and then also access to these newer technologies within within the U.S. and Europe and Japan. So with more of a focus on those new technologies.
Other questions about the background portion of Dr. Holtzman's talk?
Okay, well, we can move on, and of course, if we, if as we get into the discussion, we can loop back to these things, and it may other slides may prompt additional questions from the beginning of the talk. Okay, so let's move on now to just the stages of vaccine product development. I'll do this relatively quickly. So this is just a schematic from a publication called Nature. It's one of the preeminent journals, and it just describes the stages of vaccine and actually drug development. There's an R&D phase, and then there's an implementation phase where the product is rolled out and its impact is monitored. If we go into a slightly more detailed view of the vaccine of the research and development phase, what you start with is basically in vitro studies to study the pathogen, identify the antigens, and to develop formulations for testing. And this is one of the areas where the new technologies have really had the biggest impact, and where the recombinant DNA revolution has really been game-changing. So instead of taking months or years to get the genetic sequence of a pathogen, it's now a trivial technical exercise. And the SARS-2 virus was identified and sequenced. And do people know what I mean when I say sequence, DNA sequencing? It's to determine the genetic code, the A's and G's and C's and T's that underlie the pathogen. So it's to basically get the fingerprint or the blueprint for the pathogen. So this is now a trivial technical exercise, and the sequence for the virus, which describes all of the proteins that it's capable of making, was released within weeks of the outbreak, the initial outbreak in China. This is like unheard of. And equally importantly, that sequence, that DNA sequence, was made available to the entire scientific community to begin the process of creating vaccine prototypes for testing using a variety of technologies that, as I said, I'll cover in the third section of the talk. And so, after one sort of sequences, or you figure out what it is that you're fighting or what you want to try and develop a vaccine to, you then start to test these vaccines in animals and in animal models that you try to use to replicate the infection and the pathogenesis of the virus or bacterial pathogen. And so, I'll just, as a side comment, I understand that there are people who do not support the use of animals in research, and that it's an ethical and moral issue for some. I fall on the line of, I fall on the side of the limited use of animals to do the things that can only be done and to treat them humanely. But in the end, if we want to have confident testing of these products that they are going to be safe for use in humans, for example, for toxicology studies, there's really no way around. It's difficult to fully replicate some of those stages of product development absent the use of animal testing. But I just wanted to acknowledge up front that I understand that it's a sensitive issue and that people can disagree about animal testing. Then when we get into the next phase, once we've now, we've sequenced the pathogen, we've identified what we're trying to make the vaccine against, we have reason to believe it will be a protective antigen or immunogen, and we've proven it in animal models, then we go into clinical testing. And that's when it starts, being administered to humans. And typically there are three phases of development. There's phase one focuses on safety. Phase two testing focuses on optimizing the dosing or formulation for use in humans. And then phase three testing focuses on demonstrating efficacy. The reality is it's different than drug development. It's fundamentally different than drug development because, as I said, you're giving this to healthy individuals. And then you have to wait to see if they are protected. So that's why these vaccine studies are very, very large, because you're looking to immunize 10, 20, 30, 50,000 people, and to wait for 100 or 200 of them to get sick, you have a placebo. So the gold standard is for the efficacy studies is placebo-controlled double-blind efficacy study. And what that means is that you've got two groups. Some of them get the vaccine and some of them get a placebo. And nobody knows who's got what in terms of those who are looking at the data. And then you ultimately unblind the trial. That is, you then crack the code on who got what, and you see where did those infections fall. You need a large enough sample of people such that you'll get enough infections to determine efficacy. And there's a whole lot of biostatistics behind this. As I said, we could spend a full discussion just on the biostatistics of efficacy studies, but I wouldn't be the guy for that. I did some biostats in my public health training, and I know the issues, but you'd have to go if you really want to, reach out to other experts for those topics. But what I can tell you is that these phases are not really, it's not quite as cut and dry. So for example, phase three testing is where you actually develop the full safety database, because now you administer it to a much larger number of people. So in phase one, you administer it to a very small number of people and you watch them very, very carefully. But as you roll it out into larger numbers of people, you have greater confidence that the vaccines are safety, but you still must document safety in your efficacy studies. And at minimum, you need typically, 3,000 to 5,000 people in a safety database to have an approvable product. Okay, so those are the phases of product testing. And I've pulled a slide here from a New York Times article that came out at end of April during the early phase of the pandemic. And this is sort of a sketch on what we would call a Gantt chart of the time it takes to develop a normal product. Like if we were in normal times and we were going to go ahead and do the academic research and then the preclinical testing in animals and step it through phase 1, 2, and 3. And then you might have to build the factory and scale up manufacturing and get approval of the product and then distribute the product. This is like a 10 to 15 year process. As that first slide said, it takes billions of dollars, hundreds of millions, if not billions of dollars. And what we're trying to do now as a community is to compress all of these phases into this kind of a time frame where we would shrink, 15 years worth of work into a much abbreviated development and testing program. Now, the way this is being approached is to parallel track things. So, if we toggle back, the reason companies do it like this is because you don't want to have to build a factory if your product fails in phase 1 testing or back in the preclinical phase, right? So, it's about, the allocation of capital. But what's happened now with this Operation Warp Speed, and I do give the government credit for this, they've been willing to put out a large amount of resources to parallel track a large number of vaccines and to give them the incentive to sort of scale up and move these things into, clinical testing and manufacturing in parallel, so that there'll be much less of a gap between proving if a vaccine is effective and having tens or hundreds of millions of doses available for distribution. Now, whether or not we can achieve this, as those of you who are squinting will see that the date that this gentleman, Stuart Thompson, was saying was, by August 2021. Now, I just saw an article in The Guardian from the UK that's projecting that one of the vaccines that's being developed that you might have heard of, this AstraZeneca vaccine, and we'll talk about it in the last phase, that they may be licensing that vaccine as soon as December or January of this year. And again, we'll talk about that in context. But nevertheless, what we're trying to do is something extraordinary. I think the scientific community is aware of the issues and challenges, and I think we've made pretty good progress using these new technologies. But we will get into the pluses and minuses of the technologies in the next section in just a moment. Actually, right now. So I gave you the sketch. I hope that was clear. I'm happy to address any questions about the stages of product development. There is a fourth stage, actually. Phase 4 testing is when you have a licensed product, and then the government says, okay, look, we'll let you license this product, but we'd like to see more data on safety. We'd like you to be able to monitor this, and so you can sell the product, but we require you to continue to gather information on the efficacy and safety of that product. And this is because some of those events that I showed you earlier, if it's 1 in a million or 1 in 2 million, you're never going to see that in a clinical trial. You could not afford to do a clinical trial with 2 million people in it. And if it's 1 in 2 million, you might not even see it based on the biostatistics. So until these vaccines are administered very, very broadly, you will not capture these super rare events. And that's what the basis of phase 4 testing is, to make sure that, even once it's deployed at scale across millions or tens of millions of people, that there aren't problems associated with these products, again, which are given to healthy people. But do people have questions about the stages of product development? I threw in that phase 4 testing because it's actually very relevant, I think.
And you want to go ahead and ask your question?
Yeah, I've heard reference to phase 2A and 2B. What is the difference between phase 2A and phase 2B?
Yeah, it's a good question. Typically, when you talk about a B phase, it's in the population of specific interest. It also is. It means you've really kind of got your final formulation and you're testing that. So phase 2 testing, as I said, is oftentimes where you'll have a study that will have a dose range. You'll do some additional dose ranging, and once you get to phase 2B, it's sort of like what you want to test, and you you start to scale it up in the population of interest. Now, in the case of a pandemic, everybody's interested, right? But you might, for example, if a disease was only in, let's say it was a disease that was only in older adults. Typically, you wouldn't do your testing immediately in older adults because older adults can be more frail. They can have problems with their responses. There's this thing called immunosenescence, where older people don't necessarily respond as well to the vaccines, although there are scientific approaches to deal with that issue. So, but typically you will do testing first in young, healthy adults. So phase 2 testing might take place in young, healthy adults. You would find your formulation, and then you might do a phase 2B study in older adults who are prone to pneumonia, or older adults who are prone to, shingles, something like that. Does that make sense?
Go ahead, Holly.
You may be hitting this again later, so if you are, I'm sort of curious as to what is the definition of healthy adults, and secondly, in the trials, were they really able to get enough people from all the different ethnic groups where there have been substantially higher levels of COVID illness and death?
So two great questions, Holly. So the first question was definition of healthy adult, and then the second was really about representation of subgroups and minorities. So I think, definitions can vary slightly, but typically young, healthy adults would be between the ages of, 18 to 40. Typically these are college kids that get rounded up for testing, and people who are willing to participate in these trials. But what's important is that they not have any underlying health conditions that would be of concern. So, for example, we are, diabetes or high blood pressure, or, even more importantly, if you immunocompromised, there's no point in being, that you couldn't use these people in sort of these sorts of studies. Now, the issue of subgroups is very interesting as well, because I think there's been a big push actually to make sure that clinical studies broaden to include more than just white men. To make sure that women are participating at in order to ensure the efficacy of products in women, and similarly in ethnic minority populations. And also to make sure that there are planned studies for infants and kids, for younger children whose immune systems may respond a little bit differently as well. I cannot give you the specifics for each and every product. I mean, you'll see when we get into the SARS-2, it is a mind-bending number of products that are advancing. And so I can't really give you the blow by blow in every vaccine trial that's underway. I know that they are absolutely trying to make sure that minority populations are represented in these studies. I can't tell you definitively how successful they've been with any given product that's in testing right now. Did that answer your question, Holly?
Dr. Holtzman, I'm interested in that phase 4 testing that you mentioned, and the ongoing evaluation and assessment of the success of these vaccines once they're licensed.
Right.
How stringent those requirements are, and whether indeed they are also monitoring safety and efficacy for some time to come. I know that it took a long time, for example, for the polio vaccine, both in the old and the new form, to be pronounced to be the silver bullet that it was alleged to be in the early 50s.
Yeah, no, it's a good question. So, what I tried to make the distinction between sort of the early clinical testing and then the phase 4 testing is this, Paul. Vaccine studies and clinical studies of any product are incredibly expensive. The amount of monitoring and the blood work and all of that, it's a super intensive process. And it's why, it's why I think we have safe and effective products. So the intensity of the monitoring in phase 4 is not the same level where, someone might be, monitored like every day for, or called every day, for the first week or month following the receipt of vaccine. It's done. It's one of the big differences. It's the difference between sort of active monitoring and passive monitoring. So what will happen is, and there's a whole system actually that's in place that the United States and CDC have. I think it's called VAERS, VAERS, the Vaccine Adverse Event Reporting System. And so what they do for phase 4 studies is they make it clear to the sponsor, to the product developer, you need to do this, otherwise we're going to pull your license. And what happens is that there's active communication to people as the vaccine's rolled out, that if you find you have any adverse events, if you find that something happens, we want you to report it. Here's the number to report it at. But they're not going to pull everybody into the hospital. Once you start immunizing tens, hundreds of thousands or millions of people, they're not going to bring them in for hospital visits like they do in the first 3 phases of testing. And so that's the kind of difference. It's more of what we call passive reporting of these adverse events. It is rigorous. I mean, it can be rigorous, but it does depend on people being, self-aware and reporting these results. But it's definitely not the same level of rigor where you would have people in your phase 1, 2, and 3 studies actively come into the clinic, being monitored for disease, having nasal swabs, testing for pathogens, et cetera, et cetera. It's more that, look, we want to know if you get sick. We want to know if you have to go to the hospital ultimately for something, but that becomes separate from the trial itself, and it's the passive reporting of those results that constitutes the phase 4 data package. Okay, now let's get to, this is the topic that this is what's really near and dear to my heart. I'm sorry, are there any other questions about the stages of product development? Okay, so let's get to the sort of topic that's near and dear to my heart, which is the actual vaccine technologies. I am a molecular geek. This is what I spent my life on. And I think it's fascinating. And I think you'll see, I only have a couple of slides, but you'll see how we start to now dovetail into the SARS-2 situation. And, again, we can start talking about specific products here. And then in the next section, I am going to have to be careful because I'm not going to badmouth anybody specifically, but I'll give you my views of what's, the positives and negatives. So this is a snapshot of the types of technologies that people use for vaccines. And so starting here, I don't know if can you see my pointer or no? So we had a question earlier, I think by Julie, about inactivated viruses. And so this is like where you grow up a big vat of the actual pathogen, and then you hit it with a chemical to kill it so it can no longer replicate. And then you inject that inactivated virus in order to generate a protective immune response. Another approach is these viral vectored approaches, which you may have heard about, including this AstraZeneca product, which is made on an adenovirus backbone. So you basically take a different kind of a virus and you use recombinant engineering to insert the protein that you want to express, the protein from your pathogen, into a benign, or what should be a benign viral vector. And then that virus is often a replication competent virus, which then can stimulate immunity to the pathogen of interest. This is really another kind of virus, is what we were talking about earlier, like with measles, the original measles vaccine, and the polio, the oral polio that you used to get on the sugar cube. These are actually the virus itself, but they've been passaged to the point where they're no longer good human pathogens. But in the case of these attenuated viruses, they can sometimes revert. And that's where you get those, 1 in a million, 1 in 2 million cases where they kind of revert back to being a human pathogen. And so, this is, there are different approaches here, but, people are, I don't know anybody, I'm not sure, there may be one company that's using this attenuated virus approach for SARS-CoV-2. Then there are the nucleic acid-based vaccine approaches that people have tried for 20 years. Actually, what got me into this whole field was the promise of DNA-based vaccines, because I was basically a molecular scientist. I focused my life on DNA, And when I heard about this, I thought it was fascinating, and I thought this is going to change everything. Turned out not to work so well. There are a couple of DNA-based vaccines, but they have not proven to be effective in humans. They work for salmon. There's a horse vaccine, but nobody's been able to get a DNA vaccine to work in humans. The new wave of nucleic acid-based vaccines, we'll talk about in more detail in the next slide, are the RNA-based vaccines. So this is where, again, you're just, what I really mean by this class, though, is that you're just providing the blueprint. Remember, DNA makes RNA, makes protein. Antibodies are against proteins. And so you're giving the blueprints for the pathogen rather than the bits of the pathogen itself. Virus-like particles are a very interesting sort of relatively new technology. This is the basis for hepatitis B vaccines. It's for the HPV, the human papillomavirus vaccines that you may have heard of for cervical cancer. And this is actually what my company works on, VLPs, virus-like particles. And then finally, there are subunit vaccines, which is basically making the individual proteins and giving them as soluble antigens. And let me go to the next slide, because this is where I have the sort of pluses and minuses of the various technologies. Now, this is a small slide, I apologize, but we'll step through this from left to right. So the nucleic acid-based vaccines, these DNA and RNA-based vaccines, which are sort of showed here, their strength really is speed. I mean, these guys are in phase 3 testing right now, and they have been for a couple of months. And this is because all nucleic acids are quite similar, while all proteins are very different. And the ability to purify DNA or RNA, if you can do it with one RNA, you can do it with all RNAs. And so there's a conformity to the vaccine development and manufacturing for nucleic acid vaccines that is not true for some of these other approaches. And so their greatest strength is speed. I mean, within 2 months or 3 months, they were in phase 1 clinical testing. So as soon as the virus was sequenced, they started making these RNA vaccines, and they got them into the clinic, I think, in April. Now, their weakness is that multiple doses appear to be required, and they may, it looks as if they may not provide the most durable immunity, right, relative to some of these other approaches. That is, you get a spike in your antibodies, but then after a while, the antibody levels drop off. There are no licensed products for these RNA-based vaccines yet. It's a new technology, and there are also issues around scalability and distribution. These are the vaccines, I think, that I don't know what was it? Was it Al, where we were exchanging questions, Stan, where he raised this issue of these vaccines need to be distributed at minus 80 degrees, and this is the case for these RNA vaccines. Their RNA is an unstable molecule, so it needs to be stored frozen at minus 80 Celsius. This is not like everything, your doctor's office doesn't have a minus 80 freezer. So there are logistical issues with some of these vaccines, but the vaccines that are being developed, there's a Moderna vaccine, there's the Pfizer BioNTech. The ones that are in bold here, these are the ones that are being supported by Operation Warp Speed, which is this multi-billion-dollar US government effort. Okay, so that's the nucleic acid-based vaccines. The next one over are the viral vectors. And this is, as I said, where you take a virus and you engineer it to become a vaccine. And there are a couple of different viruses that people are using, but the ones that are really well known are these adenoviral-based vaccines. So adenovirus is a common virus. It causes a very mild disease in humans. And so people are using this to deliver the SARS-2 antigen to provoke an immune response against SARS-2 using an adenoviral vector-based approach. In this case, I think there's one of their strengths is that they may provide single-dose efficacy. Because it's a replicating virus, it may be able to provide protection with a single dose, although that's yet to be proved. And then they are often quite scalable. You can grow up a lot of this. You don't need to give a lot of a replicating virus. This is, in order to provoke an immune response. The weakness is really immune interference. If you have antibodies to the adenovirus already, it may not work in you because you're able to shut down the adenovirus. And so people are using like chimp adenoviruses that we haven't seen and other kind of clever approaches to try to use this vaccine platform. And then there may be concerns about safety in immunocompromised individuals because you're giving them a replicating virus. But these are the best example of this is this AstraZeneca is developing a vaccine that is manufacturing a vaccine that was developed at Oxford University that uses I think it's a chimp adenovirus, but I can't remember actually. And then the J&J, the Janssen vaccine product, which is another adenovirus. There's a set of vaccines that are being developed using recombinant proteins that are expressed and purified using just basic recombinant DNA techniques. And there's a lot of products that have been developed with this technology. We know it's safe because there's no virus. You're just getting a protein. These are scalable technologies. The weaknesses here is that we probably will require multiple doses. Sometimes these need immunostimulants. Your body doesn't like to react against soluble proteins. And so there may be a requirement for strong adjuvants. And then there's some issues around the stability of the protein, which I'm not going to go into. But there's a couple of companies that are working on this. Sanofi is probably the biggest company, but Novavax is another one that's been in the news a lot. And they have a recombinant approach to SARS-2 that's in the clinic and is moving into phase 3 testing. And then on the right are the virus-like particle technologies. These are also very good approaches. I mean, I think obviously I'm working in a company that's using VLP technologies, and so here's that bias. But there's the great, HPV is a phenomenal vaccine. Hepatitis B is a great vaccine. There's a reduced need for adjuvant use because your body sees the virus-like particle as a threat, and so it reacts to this because it looks like a virus, and so it attacks it like a virus. And typically these things have been very good at boosting and providing durable protection. There are some challenges with the manufacturing of these because it's a complex. Making these large virus-like particles and making them reproducibly, at scale is a challenge, and that's sort of I think something that we think we've solved with our company's technology. But that's right now the furthest along there is Medicago. They're a company that makes virus-like particles using plants. Okay, so that was the deep dive on the vaccine technologies. And I think we can probably just flow straight into the SARS-2 section because I'm noticing the time and I do want to make sure I give enough time for questions and answers at the end. And there's still a couple of important topics for me to raise so that we can get to the Q&A. Okay, so I'll go through this a little more rapidly, but so this is now that spike protein, and this is what everybody is trying to attack. This is what I think of when I think of the SARS vaccine efforts. This is the spike protein. It's a fusion protein that the virus uses to get into the human cell. And it docks through this RBD portion of the molecule, which is the receptor binding domain. Think of a lock and key. The virus attaches to your cell. There's a sort of a mousetrap mechanism that allows it to push its way through the membrane. And that's this whole spike protein. And it's just shown here in various structural formats, either from the top or from the side. And I recognize this might not be super clear to you, but it's a trimeric structure, and it's an impressive feat of evolutionary engineering based on the virus, right? I mean, these viruses are pretty clever. You've got to really respect them. So this is just a snapshot of how many vaccines are in development. I mean, this is unprecedented. I've taken this, I think, from the WHO and BBC. I've also got a link here. I'm happy to forward this slide deck to you guys as a PDF if you want, if people want. But there's a couple of different websites you can go to if you want to, follow this. If you want to obsess about the progress, you can go on a weekly basis and look at the progress for some of these trackers that are online. But this is an unbelievable amount of activity, never before seen, I think, in history, to have this many vaccines in testing and development. Now, what about that issue of safety and this issue of vaccine-enhanced disease? So one of the things that you're likely to hear about if you're really paying attention is the possibility that a SARS-2 vaccine could make the outcome of an infection worse. Now, this is different from an adverse event associated with vaccination. Because it's not that if you got the vaccine or your arm hurts, there's different sorts of, it can be, vaccine adverse events can be as mild as a headache or a sore arm, or it could be as bad as getting yellow fever. But what I'm talking about here is something a little different. What happened in the 1960s with RSV, respiratory syncytial virus, is that they used, so back to Julie's comment earlier, what they did was they grew up respiratory syncytial virus, they inactivated the virus, and they gave it to a bunch of children. Unfortunately, what happened was the vaccine was ineffective. And not only was it ineffective, but when it was given to naive infants, i.e. a child who had never seen the RSV virus before, when they got their first infection with that virus, it actually was worse. It made the infection worse. So, 2 children died. It was out of about 100 or 150 kids that were immunized. 2 kids died, and, dozens were put into the hospital. So when you looked at them compared to just what would happen if the same number of children, like of the placebo group, if the same number of children just got RSV infections, the outcomes were much worse. And this has been seen in a couple of instances most recently with dengue vaccine for reasons that are slightly different. But the possibility that the vaccine could make things worse is something that people are keeping their eye on very, very carefully. Now, I will tell you that we're thinking about this very hard from our own company's perspective. There is no hard evidence at this time of vaccine-enhanced disease, but people are looking at this very, very carefully in animal studies and in non-human and primate studies, and also in the early clinical trials to make sure that you don't see more people getting sick in the vaccine group than the placebo group, right? And that's the whole purpose of these clinical trials. Okay, now I wanted to throw this out here because this is a weird thing, and I know you guys. I mean, you're Unitarians, and you think a lot about ethics, and you think about deeply about societal relationships. And so I started early on about the Jenner experiment, where he actually basically did a human challenge trial, and it was wildly unethical because he didn't really make it clear to the people what was going on, and they just sort of went along with it because it was in the 1700s or 1800s, and so this is a little different. But this concept of human challenge studies has been used. In fact, we used it at the Gates Foundation, or we supported studies like this for malaria, because if someone gets a malaria infection, you can cure them with anti-malarial agents. And so as part of the vaccine development efforts that I was supporting on malaria, was one of the areas I worked on, we use these human challenge studies. They've also been used for things like salmonella and influenza. And what you do, again, is you immunize volunteers. Some get vaccines, some get placebo, and then you intentionally infect them with the pathogen. And this reduces the time and expense required to determine if a vaccine is effective, because you're no longer guessing about exposure, right? That's the difference between vaccines and drugs. It's not like someone has, diabetes and you give them a drug and you see how it affects their diabetes. You have to immunize people and wait for them to get sick. And that's why vaccine trials take years, or at least, months, minimally months in this case, because we have so much infection with the pandemic. But in this case, exactly that everyone is getting exposed to the infection. And so it does pose ethical challenges for SARS-2, because there isn't a simple treatment available, although there is now treatment with dexamethasone, these steroid treatments. And in certain age groups, the risk is relatively low. So if you're a young, healthy adult between 18 and 40, and you don't have diabetes and you're not obese and you don't have that, the chances of you getting really sick and dying from SARS-2 is actually quite low. It's, I don't know what the number is, but, probably 1 in 100,000 or something like that. But still, it's not a zero risk. But the UK is now saying they're getting ready to launch human challenge trials, and the US may very well follow suit. In fact, there's an advocacy organization that was established called One Day Sooner to try to help through this sort of altruistic process, if you're young and healthy and willing to get put into a challenge study, to try and figure out whether or not these vaccines work and get them out there faster. You can look into that yourself. We can talk about, your thoughts about this and whether or not this is ethical or not. It's just an interesting thing I thought you might want to know about. Now let's get into sort of more of the nuts and bolts. And this is where I think the rubber hits the road, or the tension in the system is. So who approves vaccines? So in the US, the Food and Drug Agency, the FDA, is authorized to evaluate the safety and efficacy of drugs and vaccines. But agency staff are supported by external experts, and most prominently, a committee called the Vaccines and Related Biological Products Advisory Committee, called VRBPAC. Now, I'm going to guess that it hasn't escaped people's attention that there's been certain, changes in how the political environment has been impacting the scientific dialogue around the pandemic, I would say until recently, the FDA was really quite well shielded from political influence. But I know that, I read the newspaper and I understand that it's a very big concern about people of whether or not they will be able to trust the vaccines that get approved. I think one thing that was very, very interesting for those who follow this very carefully. So the VRBPAC committee just met like last week, I think, and you can actually go online and search and see they record these things. They're publicly, you could have sit in and listened to the actual discussion with the experts. But what happened was that the FDA, Steve Hahn of the FDA, the commissioner of the FDA, what he did was he actually ended up pushing out these guideline documents to the committee as a pre-read. Now, because remember, what the administration wanted was really to have the vaccines out now, in advance of the election. And the scientific community, in fact, product developers, we don't want to rush these products out. We don't, despite the black hat that the pharmaceutical industry sometimes gets painted with. I mean, the core mission of the pharmaceutical industry is to provide medicines and drugs and vaccines that help people's lives. I'm not saying that the pharmaceutical industry is completely blameless. There's a lot of bad things that have happened in the past. I think the opioid epidemic is a glaring example of that. But I think the vaccine guys are. I hate to say it, but we're the good guys, right? I mean, people aren't really, they're not making a ton of money on vaccines, and they're certainly not trying to give kids autism. And we want to be very, very careful and thoughtful and safe with our products. But so I thought it was very, very interesting and important that the pharmaceutical industry, really supported the sort of extension of the evaluation and the safety testing, and they aligned with the scientific community and the FDA to say, no, we're not going to rush these vaccines forward. We're going to make sure they go through testing, and we're going to make sure that we have enough time to know that there aren't these vaccine enhanced disease issues and all of that. So anyway, I think that's a bit of good news, I think, and one, we'll get to the debate in a minute, but I think that this is something that people are acutely aware of in industry and within the FDA at this point in time. Now, other countries have their own regulatory bodies, and in the areas of the world that I worked in through the Gates Foundation, the WHO is really relied on for advice about what products are safe and whether or not the data from the clinical trials and the safety data justify the use of vaccines. Now, I will tell you, this is all risk-benefit, right? I mean, if there's a mild disease and there's a risk of using the vaccine, people aren't going to use it. But we're in the middle of a pandemic, so there is a legitimate discussion to be had about how quickly to roll these vaccines out. What is the margin of error for safety versus, the hundreds of thousands of people that could die if we have long delays in the rollouts of these vaccines? And that is a genuine ethical issue. Okay, so now moving toward the end, everyone is an infant in the eyes of the SARS-CoV-2 virus, and we need billions of doses of vaccine to protect everybody on the planet. And this is a big, big deal, right? I mean, it's not like you just turn on the tap and, these things this is a super time and labor intensive and expensive process. And so, sometimes people who don't understand just how much goes into the development of vaccines, and that's why I went through that whole background, think like, well, why can't we just have the vaccine now? There's a pandemic. Well, I'm sorry, it's just not that simple. It's not that straightforward. And the economics, as I said, don't support the maintenance of excess private sector capacity. We can't have companies, they just can't spend hundreds of millions of dollars on manufacturing plants and wait around like, well, maybe there's going to be a pandemic, 5 years, 10 years, 25 years from now, it just wouldn't work. And so this is where governments have to step in, and the Gates Foundation has to step in, and philanthropies have to step in to provide this kind of capacity as an insurance policy for the world. And even with that, there's the questions about this last mile problem of distributing vaccines, particularly vaccines that need to be stored at minus 80 degrees. So we're not out of this. On the one hand, I want to leave you with the message of hope that the technologies will deliver a vaccine for sure, but we're not out of it yet. And the first vaccines may be challenging to deploy. So then my last slide for the presentation is this. So who should get the vaccine first? I mean, I spent a lot of time, I spent years at the Gates Foundation on this issue of this pandemic influenza preparedness framework, which was a global framework. We supported WHO to look at supply and demand and try to figure out where should vaccines be stored. And the solution was put it in Singapore, put it in Panama, put it in small countries so that even if they take enough for their own population, you'd still be able to, they wouldn't take it all. Because what we're seeing now is the worst of all situations with vaccine nationalism and a sort of destruction of cooperation and people trying to buy up all the vaccine first. And frankly, the U.S. has been driving some of this. It's very disappointing for me to see us kind of abandoning, global solidarity in favor of, just grab everything. But to that end, we are spending the money. We certainly deserve to be at the front of the line and get vaccines. But I'd like to see these frameworks used to make sure that all countries get some vaccine to make sure that they can continue their functioning. But so this was, for the pandemic influenza preparedness framework, it's a complicated story that I don't have time to go into, but because of tensions in the global system around H5N1, do you guys remember back in 2004-05, bird flu, when everyone was worried that, this bird flu, H5N1, which killed 50% of the people that got infected, if that virus went pandemic, we were going to be in a world of hurt. And the countries that were at the center of that, at the center of those outbreaks were like, we're not going to share viruses unless we get vaccine back. And so basically there was an agreement to share 10% of global capacity with these low-income countries like Indonesia, for example, that was experiencing these H5 outbreaks in return for sharing the virus, which is what you need, in order to generate the vaccines. And countries basically were going to be able to decide for themselves who should get access first, doctors and nurses, critical governments, vulnerable populations, who gets the vaccine first is really ultimately an ethical decision. Do you give it to old people first because they're more likely to get sick? Do you give it to doctors and nurses? And that's really, going to be a question about, for individual countries to decide. But hopefully we'll be able to generate enough vaccine quickly enough that these decisions don't have to linger, and that we'll be able to get vaccine out. But it will take years to immunize the global population, right? It's not going to be something that we get a vaccine in December or January and we're done by February. I mean, just the logistics. Think about the logistics of immunizing everybody on the planet. Okay, so those are some things that I thought would be good to sort of set up for a discussion now. I want to thank you for your attention, and I'm very happy to answer any questions on any of these topics to the best of my ability.
I'll go first. Is there any possibility that the virus is going to mutate before we get vaccines out that are going to be able to stop it?
It's a good question. So I think that the answer is there is a possibility of that, but it's a very low probability based on my understanding of the virus itself. So when you, I'm not a card carrying virologist, but I've hung around with enough of them and I've thought about this pretty deeply. So when you think about a virus like HIV, it is the world champion of evolution. When you get infected with a single HIV virus, very, very quickly, you have an entire universe of viruses within you. It's so prone to change that your immune system can never get on top of it, which is why it does what it does. In the case of the coronaviruses, they are RNA-based viruses and they do mutate, but they mutate at a much, much lower level, and they mutate slower than flu. So HIV is the world champion. Flu is much less capable of mutating, but it does evolve, and that's why we need to keep getting flu shots every year. These viruses drift. With the coronavirus, there isn't a lot of data. There are viral variants out there, but the speculation of the scientific community is that if you have a good vaccine that provides responses to multiple, what we call epitopes on this spike protein that I just showed you, that the virus will not be able to mutate away from that structure, because as that structure changes, it then loses its ability to function. It loses its ability to get the virus into the cell. So I think it's something that the community will be looking at, and they will be doing that by carefully sequencing new viral outbreaks, continuing to sort of collect information on the underlying genetic structure of the virus. But I am not concerned today that the vaccines that are under development will become ineffective over the course of the pandemic. But frankly, only time will tell for certain.
This is Julie again. I have another question. Can you elaborate a little bit more about the vaccine enhanced disease? Is that caused by a person's immune response to the initial vaccine, or and it being exacerbated or strengthened?
Yeah, it's caused. So you're exactly right. It's caused by a combination of the immune system's being primed against the pathogen, but primed in a way that generates non-functional immunity. So what happened in the case of the RSV situation that I told you, and it's very relevant here, I mean, it's still relevant here, is that when they grew up that virus and killed it with chemicals so that it was no longer replication competent, they also damaged the proteins on the surface of that virus. And so what ended up happening was the kids mounted an immune response, but it wasn't a functional immune response. It wasn't a neutralizing immune response. So in other words, it wasn't capable of binding and neutralizing the virus. So what happened then was you had a primed immune system, and I didn't show you the sort of the time frame against which you recall memory, but you get better the second time you see the pathogen. You make more antibody, you make it quicker. And so what happened was that when the virus actually infected these children, the virus started to replicate. The immune system saw it and started to generate antibodies, but it generated a lot of non-neutralizing antibodies. And those antibodies are capable of binding the virus, but the virus continued to replicate, and you had large immune complexes that then get deposited, and that leads to sort of cytokine release and cytokine storms and tissue damage and inflammation and a bunch of sort of bad things that are sort of related to autoimmunity and inflammation. So what's important is that the immune response be a functional immune response. And in the case of the RSV in the 60s, the inactivated viral vaccine that was generated in the 60s, that was not the case. The kids had an immune response, but it was a bad immune response. It was a non-functional immune response. And so when they saw the virus, so they needed both things. They needed to be immunized, and then they needed to get infected. But in the case of the pandemic, pretty much everyone's going to ultimately get this virus, or a vaccine, right? I mean, it's clear this is a human virus now. And so, and that's the same thing with RSV. Not everyone is going to get malaria. If you don't go to a malaria endemic area, you're not going to get malaria. If you don't go to West Africa, you may never see Ebola, but everyone's going to see SARS-CoV-2 by the end of this pandemic, because it is a respiratory virus and it is a human virus. So you needed both, Julie. You needed the bad immunogen and then the infection.
So, okay, so can I ask a follow-up question then? I know there are, this goes back to my original question. The Chinese vaccine is being widely distributed, hundreds of thousands, maybe millions of people already. I have a friend who lives in UAE, and all of her friends are getting that vaccine. So is there a risk that now there are going to be millions of people in the world who are going to have this vaccine enhanced disease?
No, well, as I said, I think it's a very low risk. So all of the non-clinical studies using these vaccines, I'm sure that those tested. I know the product you're talking about. I forget if it's we could go back to the slide, but maybe from CanSino, it may be from another company there. They will have tested that, and they will have shown in early studies that there wasn't a problem for their product for enhanced disease. Now, I think the reason why I'm being a little bit cautious with my language is the other element of this is actually people's immune systems are a little bit different. Not everybody has exactly the same immune system. And so I can't tell you that there isn't going to be someone whose immune response and the nature of the antibodies they generate, might not be this rare one in a million, where they do worse from getting a vaccination event. What I can tell you, and what I certainly hope anyway, for the Chinese people that are getting the vaccine, is that they've been tested as carefully as the FDA will be looking at this issue. I think the answer is yes. I will tell you that most of the concerns I think that I've heard about this, the potential for vaccine-enhanced disease, really come from this a different coronavirus that people studied, a feline coronavirus, where they thought they saw this happening in cats in academic studies. I'm putting it out there as a legitimate hypothetical risk. I think it's a low risk, and I can't speak to how carefully the Chinese vaccine safety monitoring has been done. I will say that China's been a little bit in the past. There have been issues with the Chinese equivalent of the FDA. They haven't been as rigorous. They've improved. They've improved enormously over the last decade or so, because you may remember these Chinese milk issues and the milk taint in China. A couple of people actually they were literally executed. I mean, they executed the head of one of their regulatory agencies for the problems that were generated. And so this is taken more seriously now in China in terms of product quality. But I can't be definitive. I don't think it's a big problem, but it is still a theoretical concern until the vaccination and the sort of, I'm sure they're doing phase 4 monitoring in China as well.
Thank you.
I think before Holly raises another question, I just want to mention that in the chat, there's a reference that Dr. Al Edwards has provided for further information about test phases and even provide some navigation tips for working in the site. Al, did you want to say any more about that site that you mentioned?
Thank you, Paul. Other than the fact that site is primarily devoted to new prescription drugs, but it does give you a good general review of the pathway for development of almost any product. The other thing I put in the chat, and Dr. Holtzman, I'd ask you to review this, but someone had asked about the function of an adjuvant.
Yeah.
And so I've put my 2 cents into the chat on that, and I'd be interested to see if you would concur on that or not.
I'm going to stop. I stopped sharing. I'm going to go to the chat for the invitation to
Okay.
Adjuvants are substances that enhance immune potential or duration of response, and generally make immune segment more powerful in generating the host immune response. Yeah, that's absolutely correct. And so this gets into the business of vaccine formulation. And what I didn't really tell you is that, and it's kind of an interesting aside in terms of the history of vaccine development. As I said, with recombinant DNA approaches, that allowed us to do a lot of things. It allows you to make insulin, by the metric ton. It allows you to, create these recombinant proteins because you can use microorganisms in cells as little mini factories. What they found in trying to make vaccines safer. So, for example, pertussis. Right. So, the people know about pertussis, whooping cough. So in the old days, we used to get a whole cell pertussis vaccine. So whole cell pertussis was a great immunogen, but it had adverse events. And certain kids had these febrile reactions. So they would go into febrile seizures in some small, some, I don't know I don't know the exact number, if it was like 1 in 10,000 or 1 in 1,000 or 1 in, but it was a noticeable enough problem that they worked to try to get better vaccines for pertussis. And so what they did was to then create these recombinant vaccines that I was talking about, where you're making recombinant proteins and purifying them. But what they realized as they did these studies was that these soluble proteins were not as immunogenic as the whole bacteria itself. And the reason for that was because of these danger signals that I was telling you about. When your body sees a bacteria, it sees other elements, some of which now have been turned into adjuvants. So the most famous adjuvant and the most well-used adjuvant is, well, one is alum, but of the modern adjuvants, the modern immunostimulants, there's a thing that the GSK uses called MPL, which is monophosphoryl lipopolysaccharide. And this is basically a part of the bacterial cell wall. And it does exactly what Al says. And so what they realized is as they were purifying away the antigens from all the stuff of the pathogen, they became less and less immunogenic, because, as I said, your body doesn't want to respond to soluble antigens, because if you pulled the trigger every time you saw a soluble antigen, we'd all be a heap of autoimmunity. And so it's this pairing of the, this is getting that balance right of having an effective immunogen with the right amount of danger signals, but not so much danger that you cause damage to the, in healthy folks. So it's an important issue. The issue of adjuvants is very front and center in modern vaccinology right now.
One other item before Holly speaks. Al, did you want to respond to that?
No, I think that's fine. Just one other quick item. When we talked about challenge trials, and we talked about the fact that there aren't good agents to fight the current pandemic, SARS pandemic. Just wanted to raise the fact with everybody that FDA did a couple of days ago approve remdesivir for treatment. Now there's some controls wrapped around that, and that is you have to be hospitalized and you have to be near or pretty close to needing a respirator in order for the drug to be used. So it's not to be used lightly for, let's say, someone that just walks into an emergency room and has a fever and a few other confirming COVID-19 symptoms. But there is that, let's say, backstop now for someone that has a very severe infection. It comes with some severe caveats as well in terms of closely monitoring your liver and kidney function. So again, it has to be in a controlled situation where people really know how to use the drug in order to get the efficacy. And as Dr. Holtzman has talked about, suppressing the bad side effects of things. One other quick thing I just wanted to mention, and I don't want to either implode or explode this very elegant discussion, but and I think many of you folks are Unitarians, so you have kind of an ethical bent to life. Remdesivir, as well as many vaccines, are supported or in part made with stem cells. And the origin of stem cells is from abortion material taken from people in the 1970s and 80s. And so there are some individuals who have, let's say, such a belief in their position on abortion that they may not take certain vaccinations based on how they're made. And just wanted to open that up, not to, let's say, steal the thunder here, but just make sure there's kind of a truth in manufacturing, truth in origin for where some of these things come from and what the origins are.
Thank you. Thank you. Holly, go ahead.
Excuse me. Dr. Holtzman, my question is slightly related to what I asked at the beginning, which was if Biden were elected, what would you recommend? I'm hoping that the CDC or someone is going to come out with some kind of recommendations, but it sounds like it's very possible that there are going to be 2 or 3 potential vaccines available. And how will the efficacy of the different ones, will the CDC say this one has a 50% and this one shows a 40%? I mean, what kind of information will we get, and what kind of information do we need to be able to make, informed decisions about not only about our own health, but in general about the health of our country and the world?
Well, it's a pretty good and tough question, Holly. I mean, so the answer that I would give to Joe Biden, if, maybe it'll sound a little self-serving, but I wouldn't ask him to come talk to me. I'd say, go talk to your experts, listen to the scientists. I mean, don't bully it through. And, that they're the ones that are closest to the data. And I think that they're, it's sort of been mind-bending to me, like watching what's been going on. But so I think that there are still good people, who can evaluate the data within the agency. So remember, the FDA is going to be the group that would look at the data to say, yes, it's safe and effective. They have these advisory committees of experts. I know some of the people from my time at the Gates Foundation who are on that VRPAC. They're incredible scientists, scholars, and clinician researchers. And so they're the ones that can evaluate the totality of the data to say, yes, this is safe and it should be approved. I think you're right. You will get data on the efficacy, but remember, there's going to be a lot of noise in the numbers, right? I mean, what happens is even with 30,000 people immunized, maybe you'll end up with, 100, 200 cases or something like that. So the biostats around this and the confidence intervals around how effective, how confident you can be about the exact efficacy of any given vaccine is going to vary. And typically what they do is it has to be something greater than 0. And then it depends on how, so it's got to be for sure, the biostatistically meaningful, but, that's where you've heard these discussions. I mean, some vaccines, they probably wouldn't license if it was a mild disease and it was only 50% effective. They might not say that that's good enough, but for a pandemic, 50% may be good enough. And so I think what you'll see is some metric for efficacy. We in clinical testing, this vaccine protected, 50%, showed 50% protection against disease. Or the other piece of this, and Al will know this because I think he sounds very, very well informed about the FDA. It also matters, what's the endpoint? Is it severe disease? Is it, infection? Is it hospitalization? And so they're not going to be able to have, it'll probably have to be against something more like disease with symptoms, or maybe hospitalizations, but, I don't know what each trial is looking at as an endpoint. So I think what you're going to have to do is you have to listen very, very carefully to the rollouts of the information and figure out for yourself. The other thing I can tell you is that certain vaccines may perform better or worse in different populations, like the elderly, right? And so, I was shocked when the AARP started sending me information. I consider myself a young man, but I'm all of a sudden I find out that I'm not. It was a shock, and so these are the kind of things where you're going to want to keep your eye on it. I mean, I think the first wave of vaccines will almost certainly be the nucleic acid vaccines and possibly the viral vectors. The protein-based vaccines will probably come in soon after that, and then people were asking about Icosavax as candidate. We just got started. We were just fortunate enough to receive a grant from the Bill Melinda Gates Foundation, and we're trying to get our product into the clinic by the middle of next year. But it's going to take us a couple of years. But the reality is, in our opinion, this virus is not going to go anywhere. It's going to evolve into a human virus that's a seasonal virus. That's speculation, but that's what I think is going to happen. And so, just like flu, people are going to probably need this vaccine for years to come. And so, we're not trying to catch this first wave of, people getting the vaccines, but we're trying to make the very best vaccine through a virus-like particle approach that would provide long and durable immunity safely to all people. But I think the first waves of vaccines will probably, they won't let them out if they're not effective, I don't think. But they may be, maybe they're only effective for 6 months, right? Because we only found out about this pathogen, 12 months ago, right? Or not, 10, 11 months ago. And, so they can't guarantee you that these vaccines that they're going to release, possibly under emergency use authorization, will provide you protection for 2 years or 3 years. The best they're going to be able to say is that these people who got immunized in this trial, it looked like, they were 50% protective. But the duration element is something I would also keep my eye on as the information comes out.
Dr. Holtzman, do you think that it kind of as an offshoot of Holly's question, will the information, can the information that comes from the CDC or the FDA be so clear that consumers of vaccine can make a choice between or among them? I mean, it begins. If I take one thing away from your presentation, it's the complexity of these issues and the ramifications of the trials and the tribulations, if you will, that the trials lead to. But I don't know whether I should use Gleam or Colgate, or Crest, if all three of those vaccines are produced.
Right.
Is there a way that I will know that?
I don't know, Paul. I mean, I think that if there are certain limitations in various populations, they'll be noted. So, for example, like these live viral vectors probably won't be given to immunocompromised individuals. The issue of, we don't have a national plan. I'm sorry to say it, but, this whole pandemic has been bollocksed up so horribly that, we still do not have a national plan. That's why I shared that article from the New York Times. Like, it's not going to be straightforward. I don't know what to say. I don't know if in the end, the US government is going to say, here's the vaccine, people on the West Coast, you get this, people in the middle, you get that. Or based on population, I have no idea what's going to happen. But I do know that I would like to believe that they won't release any vaccines that aren't safe and at least partially effective. I don't know whether or not, I don't know who's going to get what first, like I said in that last slide. I don't know, I don't know whether or not they're going to allow the elderly to be the first to go, or if they're going to do doctors, or I don't know if they're going to roll out all the vaccines at once. I know we've made investments, the US government's made investments across the board, right? They've spent, I don't know how many $1 billion to commit to pre-purchase of vaccine from AstraZeneca, from Pfizer, from Johnson & Johnson. So there is going to be a bit of chaos, I think, and I don't know any way around that right now.
All right. We probably need to wrap up because we've gone a little bit past time, but the interest is here in continuing, I'm sure. Are there other questions before we?
I think I got time for one more question. It's really been an absolute pleasure to chat with you. I hope what I presented was clear and helpful. I recognize it's a complicated issue. I tried not to be too doom and gloom or too overly Pollyannish, and to try to give you at least what I see as the straight, the best I could do to sort of lay out the facts as I see it, with whatever commentary I've shared. I would be optimistic. I am optimistic, and that we will have a vaccine. So I want to leave you with some optimism. I don't think it's coming tomorrow. I don't think we'll be all fully immunized. I think we're in for a rough ride through the fall and into winter. That's the unfortunate reality. But I do think you should have cause for optimism that this is a pathogen we can tackle. We will have vaccines. They will be safe and effective if we let the scientists if we let the science lead. Anyway, any last question? I'm happy to.
Any last questions? And anybody who's interested in the slideshow, put your name in the chat, please, before you sign off. And just let me know if you're interested in that, and we'll see that you get that. And this recording of this talk will also be available should you want to hear it again.
One quick question. What percentage of the population will we need to have herd immunity?
Yeah, that's a great question. Again, it's an unknown, Holly, because it differs for each virus. So for example, with measles, to really disrupt transmission, it's such an effective virus, and the way it hangs in the hand as air as an aerosol, you need to have like 90 or 95% of everyone immunized in order to sort of break transmission. For flu and probably for other viruses, it's a little probably less than that. You may have heard originally the estimates were probably like people were thinking 70%, right? Because that's about a good number. But the truth is that community immunity or herd immunity, it isn't really a digital thing. It's not like, oh, you hit 70% and then there's no transmission. You're still going to have pockets if you have a large area of people that are non-immunized. I've heard people, and it may be wishful thinking, suggest that some of the transmission dynamics in the New York metro area suggest that community immunity may actually arrive at some lower level. But I'm not enough of an epidemiologist, and I don't know if I've looked at that data carefully enough to sort of sign off and say, oh yeah, it's more like 40% for SARS-2. It's a weaker virus, it doesn't transmit as effectively, and it's 40% is what we need to hit. I think what I can say is we're nowhere near that, right? I mean, this is the idea that we're going to just let the virus run wild and nobody should be taking care of wearing masks or trying to use public health measures to limit transmission. That's pretty much foolish. I think Sweden tried that, didn't really work, and I think the answer to your question is I don't know. If I had to guess, I'd say in that 60 to 70% range, you'll start to see transmission slowing. And so between a combination of the maybe, 10, 15% of people who've already been infected in our country, and then a wide rollout of vaccines that even if they're partially effective, hopefully transmission dynamics will change and we'll be able to monitor that if we have testing, if people, if we can get our act together as a country, on the public health side. So
I would think it'd make it even more complicated because it looks like there's some people who may lose immunity once they've had immunity. So how do you even think about herd immunity if that's going on too?
Well, it's very complicated, and there's super smart, epidemiologists that make models around this. The one thing I can say though, Stan, is that, back to the sort of first principles of immunology, one of the reasons why it's probably worth it to take some of these, first wave vaccines that may or may not be perfect. No vaccine is perfect. There's no vaccine that provides 100% protection for all people forever. But some of them come close. I mean, things like measles and yellow fever, these are really great vaccines. But once you've seen a pathogen once, or once you've been immunized and have memory, you're better able to fight the pathogen. Your immune response comes back faster. So even if you get an infection, you should be able to fight it off better, assuming there's none of this other problem that I raised before about this vaccine-enhanced disease. So even a partially effective vaccine, if it triggers memory, if you have immune memory, when you then get the virus, you should have a less severe case. The problem right now is we are all infants in the eyes of this virus. Everybody's getting a primary infection, and that's that's like the very worst you can get. And so for people who are older or people who have underlying health conditions, it can be very serious. For some subset of younger people who may have this, particular weird immune system that creates a cytokine storm, doesn't know how to fight it off, because there are young people who are dying from this, right? It's not very many of them, but it's probably a subgroup of people who have, subtle differences in their immune system that just, it's tough luck. They drew a short straw and they're not able to fight off this particular virus. But I mean, the issues of community immunity, herd immunity will be looked at very carefully. But, get your flu vaccine if you haven't already gotten it, read the papers. In Fauci, we trust, don't and stay, be careful what you see on Facebook. I guess. And my wife works for Facebook, so
Uh-oh.
don't tell her I said that. Scratch that from the recording. No, anyway, look, it's been a pleasure. I want to thank you all for your attention. It was really nice to meet you all. And of course, stay safe, stay healthy, treat each other with compassion, and I'm sure you will. I know it's hard for everybody. This is a very strange year we're living in, and but I'd like to believe that I am an optimist, and I'd like to believe that 2021, by next summer, maybe next fall, we'll be in a very, very different place with the pandemic.
Thank
Your
you.
mouth to God's ear.
Thank
you.
you.
Thank you.
Thank
Bye,
you very
everybody.
much.
Take
Yeah.
care, everyone.
Thank you.
Pleasure.
Thank you.
Thanks, Paul.
Make sure you voted.
Yes.
Yes.
Amen.
Yes. Thanthank you, Paul.
Thank you all.
Thank you.
Thank you. Okay.